Welcome to the all viewers and well-wishers of the world. The purpose of my lecture is learn the knowledge from the word and return the knowledge to the word. Motto of my life is learn, earn and return. I have learned a lot, earned a lot, now to return a lot to the word. The topic of today is antibiotics or antimicrobial resistance. Introduction. Since first use of the antibiotics in 1930s and 40s, bacteria adopted and developed the mechanism to escape antibiotic effect and to survive. With time, new antibiotics were developed to overcome this resistance which the bacteria have developed. Since 1990, new antibiotic development has fallen and bacterial resistance has increased. There is much more antibiotic have been developed to overcome this resistance. Antibiotic resistance is responsible for countless human deaths, and resistance is mediated by the chromosome, plasmid and transposons. Here one can see exposure to the bacteria occurs and infection occurs and the bacteria spread. One bacteria multiplies into many bacteria, but as the drug treatment or the antibiotic was given, these bacteria just died and the person is healthy again because of the antibiotic which has been used to the multiplying bacteria. Now, drug-resistant bacteria are those which have got some gene, some plasmid, some chromosome, and here one can see the red part of that genome of the bacteria, and these are capable of living and surviving even in the presence of the antibiotic. Now one can see the bacteria multiply and the bacteria continues to spread and the person remains sick and dies because of the bacteria which are not responding to the antibiotic. So this is the resistance to the antibiotic by the bacteria because they have got certain gene in them and they do survive and multiply. Now, antibiotics are used in human and veterinary medicine to control and to treat infectious diseases. Their use and misuse of the antibiotic cause emergence of the resistant strains of the bacteria. Antibiotic bacterial resistance in animal is not a risk to the animal but also to the effect to the humans because when transmitted as foodborne contaminants in the human, there is bacterial resistance. Understanding how bacteria become resistant to antibiotics and factors responsible for resistance are extremely important. Now, the definition is drug-bug interaction, means drug and bacteria interaction. Resistance of the microorganism to an antibiotic or to an antimicrobial. Relative or there is complete resistance of the antimicrobial, as the name is. Complete means complete and relative means relative. Against a previously sensitive microbe. Previously microbe was responding to the antibiotic, but now it is not responding. There is increase in MIC, minimum inhibitory concentration of the drug, and a bacterium carrying several resistant genes, multi-resistant or a superbug is just resistant to many antibiotics. So one can see the resistant bacteria and their weapons, and they can just planning to kill the host, and these are all the superbug and the resistant pathogens. Now, many bacteria become resistant to all antibiotics, including the vancomycin, which is considered to be the very strong antibiotic to kill these bacteria. An antibiotic which is being used as a last resort to kill these bacteria. There is cross resistance versus multiple resistance. Cross resistance means there is single mechanism by which the bacteria become resistant, and that is found in the closely related antibiotic, and the bacteria are just resistant to one antibiotic which contain beta-lactam, and they are resistant to all the antibiotic which contain beta-lactam. Now, multiple resistance is the multiple mechanism unrelated antibiotics or bacteria by the different mechanism, and they just to avoid the antibiotic action, and they become multiple resistant. So here one can see the cross resistance, that is the bacteria just when they enter into the host cell that is being guarded, and it does not allow the antibiotic to enter into the host cell, and that is called the cross resistor. But here in this diagram, the bacteria get different way to get into the antibiotic, that resist the different antibiotic to get enter into the bacterium, that is by the efflux action they kick out the antibiotic, and permeability does not allow the bacteria to enter, and they alter the target sites for which the bacterium has been fired. And similarly, there is the pore influx or pore permeability, and the plasmid with the antibiotic resistant genes, that is just to combat the antibiotic, and they just destroy the antibiotic and does not give the chance for the antibiotic to act on the bacteria. So this is the multiple resistant mechanism, and here in cross resistance there is only one mechanism, that is just the antibiotic cannot enter through the foreign molecules into the host cell. Now, the certain key terms which are being used in antibiotic resistance, that is, antibiotic is a drug that kill or inhibit the growth of the microorganism. Resistant is means an antimicrobial will not inhibit bacterial growth at clinically achievable concentration. Susceptible, an antimicrobial will inhibit the bacterial growth at clinically achievable concentration. Susceptible means the bacteria that is being killed by the antibiotic and that is sensitive to the antibiotic is the susceptible. MIC, minimum inhibitory concentration, the lowest concentration of the antimicrobial that inhibits the growth of bacteria. And similarly, the MBC is the minimum bactericidal concentration of an antimicrobial agent that kills the bacteria. Breakpoint is the MIC that is used to designate between susceptible and the resistant bacteria. So, acquisition of the resistant in the bacteria is by two ways. One is by the natural way and other is by the acquired way. So the first, the natural resistance means it is also called inherent resistance, that is the all bacteria cannot be killed by one antimicrobial agent. Bacteria naturally or inherently resistant to an antibiotic because of certain changes in the different, that is just like one, the different cell wall. So the organism lack a transport system for an antibiotic, means the porin does not allow the antibiotic to get enter. Organism lacks target of the antibiotic molecule RNA, and the antibiotic fight to kill an RNA is not lacking in that antibiotic. And in gram-positive bacteria, cell wall covered with outer membrane, and this outer membrane is a permeability barrier against antibiotic, and it is mostly seen in gram-negative bacteria, so they are resistant to the many antibiotics. Now, one can see the natural resistance, that is some intrinsic resistance, mean the antibiotic which act on the cell wall, the bacteria just lose the cell wall and the no cell wall is there, so antibiotic cannot act on the cell wall and that is not capable of killing that bacteria. Similarly, there is reduced permeability of the membrane which does not allow the antibiotic to get inside. Similarly, the porin molecules are closed and does not allow, if just by chance the antibiotic get inside the efflux pump, just kick out the antibiotic, so the antibiotic does not have the time to work. So this is the natural resistance which the bacteria get developed by to mean to survive their existence, that is by the natural resistance. Now, an arrow can only be shot by pulling it backward. So, to get anything forward, you have to pull it backward. When life feels like it is dragging you back, it is actually you are launching to something great. Just focus and aim. So, just dragging backward and pulling backward does not mean that you have just lost your aim and target. Now, the acquired resistance, that was natural, now this is acquired resistance. It is of two types, vertical evolution and horizontal evolution. Vertical evolution is vertical and horizontal evolution is horizontal. And one will feel easily to understand that vertical evolution is the mutation or selection pressure, and the horizontal evolution is conjugation, transduction, and transformation, by which the bacteria get or transfer certain resistant genes to the other bacteria, and they do survive because of that genes which have been transferred and transmitted to the other bacteria. Now, acquired resistance is acquired by the mutation of its DNA or acquiring resistant DNA or genes or DNA from the another source. It results from the changes in the bacterial genome. So the change in the bacterial genome is responsible for this acquired resistance. This occurs by two genetic processes. One is vertical evolution, I have told you, vertical mutation and selection pressure, and horizontal evolution means horizontal gene transmission and exchange of the genes between the strains and the species. Now, acquired resistance, one can see the exposure to the bacteria occurs and the bacteria do die because of the effect of the antibiotic. But some of the bacteria, they acquire the genes, as one can see, the red color, and now they are capable of resisting the antibiotic which was capable of killing these bacteria. Now they are able to survive, they multiply and make sick the person. Vertical evolution, it includes mutation and selection pressure. Mutation is spontaneous mutation or the change in the bacterial chromosome that causes change in the DNA. It results in the target of the drug in bacteria, so drug does not bind. Change in the membrane, so drug does not penetrate into the cell. So these are the changes which are the mutation, and that is just does not allow. Here one can see the mutation. It is a change in a gene or chromosome. The bacteria which was not having mutation was able to die when the antibiotic was applied. And here the bacteria get mutation and it is able to survive, and here it is just enjoying its presence and continue to reproduce and produce offspring that resist antibiotic use to treat it as it was here. And here one can see a kind of bacteria, there few bacteria among this population, that non-resistant bacteria exist in this population, and here one can see the mutation in the DNA of this bacteria, and that is in the presence of the drugs. This mutant bacteria which have got the mutation in the DNA with red color mutation gene, they are able to survive and they can multiply, and the drug resistant bacteria multiply and live happily. Mutation is a change into DNA that can sometimes cause a change, and here one can see the antibiotic getting into and just having the effect on the DNA, and here that is just affecting the DNA, and the rest of the bacteria is going to die. And again, the antibiotic is just going to affect the DNA and going to die. But here the resistant gene is there and that have no effect of the antibiotic, and antibiotic just pass it and the bacteria is able to survive. So that is the mutation one can see. In selective environment of the antibiotic, wild type, which are non-mutant, are killed and resistant mutant are able to grow. The resistance is by the Darwinian biological principle of survival of the fittest. In large population, few cells can survive in the presence of noxious substance, and they are able to defend the action of the antimicrobials, and the susceptible organism will be eliminated. And here one can see the selection pressure, the bacteria, these are the wild type non-mutant, and these are being just able to multiply, but that when some antimicrobial is coming, that is able to kill the wild type bacteria, but some of the bacteria get the mutant gene in the DNA, as one can see the red color mutant gene, and these are capable of resisting the antibiotic which is being used to kill them. And here one can see they can multiply, and these are drug resistant bacteria which multiply and thrive and are able to cause the infection. Similarly, here in this picture, the harmful bacteria colony is there, and these are just being killed by the antibiotic, but one of the bacteria is able to resist the effect of the antibiotic which has been applied on this dish, and as you can see the blue color, but this is able to resist, that is the mutant bacteria, and here one can see the antibiotic resistant cells, the red color, survive and proliferate. Now here is selection pressure, and how it works. Selection pressure, one can see the increased prevalence and dissemination of resistance is an outcome of the natural selection and should be viewed as the expected phenomena of the Darwinian biological principle of. Here one can see the bacteria of different kinds are here, and as the antibiotic is being given, the wild type of bacteria here one can see these are not able to withstand the effect of the antibiotic and they do die, and these which are able to resist the antibiotic, these are able to remain, and here even when the antibiotic is applied, they are capable of living and thriving, and now they start multiplying and able to infect and cause infection in the presence of the antibiotic which was given to them. But as they are resistant to that antibiotic, they are not going to have the effect of. Now the selection pressure, and now we are going to see that what is horizontal evolution. I have told you the horizontal evolution It is also called horizontal gene transmission or HGT. It is exchange of the gene between the strains and the species. It occurs by three ways. That is, one is the transformation, the other is transduction, and the third is conjugation. I have just described this in detail in case of the topic of the genetic transfer of the material and bacterial genetics. One can have that in more detail and to understand that. But here again, I'm just trying to make it understand for you. So that is transformation. During this process, genes are transferred from one bacteria to another as the naked bacteria. Here, the bacteria which do die, it has released its plasmid DNA and chromosome, the genetic material, which is being taken up by the another bacteria, and these are having the resistant genes, and these are just incorporating with the chromosome to make this bacteria resistant to the antibiotic. So this is called transformation. After taking the wild genetic material from the dead bacteria, these are going to resist. The next is transduction. Here in transduction, one can see that the genetic material is coming, but this is how that genetic material is coming, that one bacterium to another via a virus or the bacteriophage that infects one bacteria and transfer the genetic material. Here one can see the bacteria being infected by the bacteriophage, and the bacteriophage takes the genetic material from this bacteria and it is going to transfer this genetic material to the another bacteria so that to make it resistant. As one can see, the bacteriophage are just attacking the different bacteria and they are capable of putting their genetic material into these bacteria and just making them resistant. Now the third method is conjugation. Here one can see it is also called the bacterial sex, that it is a particular kind of the circular DNA called a plasmid which replicates independently of the chromosome, and this is capable of just getting from one bacterium to the another via a sex pilus. It contains a resistant gene which is going to be transferred to this bacterium to make this bacterium resistant. As one can see that this plasmid, it just opens up and it is being transferred via this sex pilus from this bacterium to the another bacterium and then incorporating with this genetic material. And here again one can see the genetic material with resistant gene is transmitted to this bacterium and to make this bacterium resistant. And the next is the summary of horizontal transfer. As one can see here, transformation takes the wild dead bacterial's chromosome and that is being transported in the form of transformation. Conjugation, that is just by the sex pilus, the genetic material is transferred to this bacteria which is not resistant previously, and here via the bacteriophage the genetic material is injected into this bacteria to make it resistant. Now here again, simplified way, the conjugation, the genetic material of this bacterial plasmid which is resistant is being transferred to this bacteria to just transfer the resistant gene here. And here transformation, the wild genetic material of the dead bacteria is being taken up by these bacteria to make it resistant. And here in transduction, the bacteriophage is transmitting the genetic material taken from this bacteria and just putting it here, and it is called bacterial sex. So the causes of the bacterial resistance are overuse or improper use of the antibiotic, prophylactic antibiotic before surgery, empiric use without knowing the which organism is there, incorrect diagnosis and the wrong antibiotics, the correct diagnosis with wrong antibiotic, and increased use of the broad spectrum antibiotic in place of the narrow spectrum antibiotic, and pediatric use of antibiotic for the viral infection. Similarly, antibiotic for the cold, flu, herpes, or tinea capitis like diseases which do not need antibiotic. So here is the summary of that in diagrammatic way. Over prescribing of antibiotic, patients not taking antibiotic as prescribed by the doctor, and unnecessary antibiotic used in the agriculture and in this farming for the animals, and then poor hygiene and lack of the rapid laboratory test to diagnose properly and to fire the proper antibiotic after doing the culture sensitivity of that. Now, continuing the causes of the bacterial resistance, patient do not complete course chronic disease and leave in the way, and for the TB and the AIDS do not complete their antibiotic course. Use in the veterinary practice, food and agriculture to have the more yield, but expanding population of immunocompromised patients are also one of the cause of bacterial resistance. Increased use of invasive medical procedure and vector resistance, lack of the quality control by the manufacturers, and the defective susceptibility assay to give the wrong reports